So you've probably had this information before, but I want to make sure we get all on the same page for it. We're going to cover just now the basics of genetic inheritance and the principles of inheritance. This is something that should have been covered in your introductory biology course. So if you look at yourself and kind of start from basic foundations, let's say you are this person. Where did you come from? How did you get into this world? Right? So normally I leave this open to questions. People answer things like, well, I got here from my parents. So we got the birds and the bees, right? Let's say this is you. Da-da-ding, da-da-ding. All right, so a person has two parents. So biologically, children are born from a male parent and a female parent. That's the male symbol here. And here's the female symbol here. And those people get together and produce this child. So that's, pretty basic. I think everybody knows that. But what is it that these individuals are contributing to that child? Well, they contribute some cells. So the male parent has a sperm cell, female parent has an egg cell. And before you were you as a recognizable person, you started off as a single cell. So genetically, pretty important and I think very interesting that we can trace ourselves back to a single cell. And genetically, there's so much of the programming, what made you, that was contained in that single cell. So whatever your parents gave you basically had to fit into that cell. They couldn't give you strands of hair or organs or anything like that. They had to give you the programming to make those. So inside of these cells, as we will see, these single cells produced from your parents, those have little bits of DNA, and your mom's DNA and your dad's DNA combined together to produce this single cell zygote. And then that zygote grew up through cell division, produced more and more and more and more cells until you had so many cells that they organized together into a human looking organism. So just fascinating that there's so much programming contained in one cell that it can make a human, control the production of all these organs and features, and also, put forward these differences among people that are so interesting. All right, so going way back, you started as one cell produced from the sperm and egg cells contributed by your mom and dad. This is how they look during the process of fertilization. So during the process of fertilization, the egg cell is quite large. That's large purple kind of magenta globe in the background is an egg cell. And typically there are lots of sperm cells that are competing for the access to the egg cell. And only one of those, in most cases, only one of those sperm cells is going to be successful at fertilizing. So one term that we're going to use is gamete. And gamete refers to sperm cell or egg cell. These are single cells, and they contain, as we'll see, they have 1/2 of the total genetic information. So your mom gave you half of your genetic information, your dad gave you half of your genetic information, and those combine together into that single cell that results, that will be the zygote. So eventually one of these sperm cells is going to make it inside of the egg cell. Sperm DNA mixes with egg DNA, and then the egg cell then genetically goes on to become the zygote after it's fertilized. So looking inside of those, we can look at these in terms of chromosomes. So these are human chromosome numbers. You would not see the chromosomes as such inside of the cells, but it's, I think, a nice way to think about the genetic information that's contained in there. And this kind of a relative size, the sperm cell would be relatively even smaller next to the egg cell. But as I mentioned, we have half of the information coming from mom and half of the information coming from dad. So you're probably familiar that humans have 23 chromosomes. And what it means is actually 23 kinds of chromosomes. So you actually have 2 of every kind of chromosome. So we have an official numbering system for these. These are just kind of cartoony right now, but your mom contributed one version of chromosome 1, your dad contributed one copy of chromosome 1, and that means that your cells got two copies of chromosome 1. You got two copies of chromosome 2, dad's copy and mom's copy, and so on all the way down to chromosome 23. Chromosome 23 is a special case. This is what we call the sex chromosome, or the X and Y chromosome, and males have an X and a Y, and so when the sperm cell is made, it would get either the X, which is larger, or the Y, which is smaller, and depending on which one of those is given by the father, that will determine the sex of the baby that's made through this fertilization event. So the chromosomes are what's combined, and chromosomes, as we'll see, are made up of DNA, and DNA, as we talked earlier, is what genes are made of. So contained within these 23 chromosomes are thousands of genes contained on those DNA, and they get combined in a contribution from dad and a contribution from mom. So after fertilization, as I mentioned, your one copy from mom, really one set of these nuclear chromosomes, one set from mom and one set from dad, they get combined together. And they're present in the cell. Again, they wouldn't look like this, but it's a good way to think about them. And they aren't necessarily hanging out together or anything like that. They're just part of this total DNA in the nucleus of the cell. And within that, you would have two copies of chromosome 1, two copies of chromosome 2, two copies of chromosome 3, and so on. So that's what's contained in your typical cells. You can actually see the chromosomes. You have to catch them at the right time. So we'll talk about this in a little more detail when we get to the topics of mitosis and meiosis, and we talk about chromosomes. There's a time in the life of a cell when it packages up chromosomes, and you can actually see them using a lab technique. We'll have a lab where we actually are able to look at some chromosomes. And this is what's called a chromosome squash, where the chromosomes are stained to have a color, and then they are pressed very firmly down so that they spread out so that we can count them individually. So normally these would be all pretty tightly packed in the nucleus, but having them spread out here, you can see them individually. And so I told you humans have 46 chromosomes. Let's see if we can count them. Conveniently, there's a dark spot on these chromosomes, which is the centromere. So that's the kind of the anchor point that holds the chromosomes together. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30, 31, 32, 33, 34, 35, 36, 37, 38, 39, 40, 41, 42, 43, 44, 45, 46. Nice. Still haven't messed that up yet. So there are 46 chromosomes. As I mentioned, they're not connected to each other, those paired chromosomes, but you'd be able to match them up based on size. So the two longest ones kind of look like this one and this one. Those might be the ones called chromosome 1. This is also a long chromosome. Here's another long chromosome. People who do this professionally would be able to line those up and tell you which chromosome is which. All right, so if we have this version of cells where the egg cell has a half amount and the zygote gets a double amount or a whole amount, or maybe just make it more clear, 2 copies of each chromosome in the zygote, or a body cell called a somatic cell. So this is most of the cells in your body have this amount of chromosomes, 2 copies of each chromosome, and only specially programmed cells, the result of producing sex cells, those are the gametes, such as an egg cell, and those get only 1 copy. So how are those produced, going from 2 copies to 1 copy? We'll cover that in detail. That's the process of meiosis. And that's shown on this cycle here. This is the life cycle. So this is what I call here the sexual reproduction life cycle. This is common across organisms. So can't really think of exceptions within the at least multicellular eukaryotes. So humans, animals, plants, fungi have this same life cycle They do things a little bit differently across different kingdoms of life, but by and large, it's pretty much the same. Where there's a set of cells that have a double amount and a set of cells that have a half amount or a single amount. And the cycle part means that it goes through, switches between these two kinds of chromosome numbers. So the double amount is called diploid. And as I said, that's the most of your cells. And the process of meiosis is this process of going from diploid, or two sets of chromosomes, to haploid, which is one set of chromosomes. So that's a process. If you're producing sperm cells or producing egg cells, the process of meiosis produces those cells. Sperm cells and egg cells are made for the purpose of fertilizing. And so if you've produced an egg cell, your egg cell can combine with a sperm cell in the process of fertilization, and that would result in a zygote, and that would end up being a new individual. So it is a cycle, it's sort of a cycle for species, but at this point, as a result of fertilization, this would be the start of a new individual. So it's not like back around again, but it's a new person after fertilization. So a sexual reproduction life cycle goes between diploid, double amount, and haploid, single amount, every time we do the life cycle. This is a full circle. This would also be known as a generation. So your parents' generation, your grandparents' generation, your generation, this is the amount of time that's spent going from one of these to the next. So some other terminology, I've mentioned that you would have 23 types of chromosomes. Those chromosomes typically have a number, so we number them 1 through 23. So I've got down at the bottom here how you would refer to them: chromosome 1, chromosome 2, chromosome 3, and so on. Not every organism has the same amount of chromosomes, the same number, but within a species, that's pretty standard to have the same number of chromosomes. And because they're paired, I've got them colored here in 2 colors for each pair, suggesting maybe the contribution from dad in blue and the contribution from mom in orange, something like that. And those pairs together, the one from mom and the one from dad together, those are known as homologous chromosomes. So homologous chromosomes are the same size, so they have the same length, they have the same centromere position. This little pinched in part here indicates where the centromere would be. And what we can't see on here, but what we would understand is that they would have the same genes in the same order. So along this chromosome, there might be a location or an address on here where you would find a specific kind of gene, and that's standard across the human species. So if you've got a gene for eye color, wherever your eye color gene is located, the gene of that same kind would be located at the same place on somebody else's same chromosome. And that's kind of important for people to be genetically compatible with each other. All right, so 1, 2, 3 on here, you could have up to however many the organism has. Zooming into a little more detail, I mentioned the genes. So you can have genes at the same location, but one of the things we find interesting to talk about early on in genetics is that not everybody has the same versions of all of their genes. So a different version of a gene is called an allele. You've probably seen before. And alleles are different versions of the gene, but still located at the same place on the same chromosome. So if a person has chromosome 1 with this gene address for gene B, they have 2 copies of gene B, and the capital and lowercase of B are indicating that they've got 2 versions of that gene. So the big B version is the big B allele. The little b version is the little b allele. And those are at the same location on homologous chromosomes. So a person can have 2 different alleles for the same gene. They can have 2 of the same alleles, little t and little t. This notation is basically what people use as we understand the gene. So if they're so similar we can't tell them apart, we'll give them the same name, the same letter, little t and little t, little n and little n, and then maybe some differences, little q and big Q. And so this would be the genetic makeup of an individual. And we typically focus on just a few genes at a time, But just bear in mind that you're looking at tens of thousands of genes in your genome. Many of them we're not looking at at any one time, and any one genetic study typically doesn't look at all the genes in the organism. Okay, so if we have these alleles present on chromosomes, then we can track a specific allele set, look at one gene and its alleles, and follow that through the life cycle. So let's say somebody has two versions of the G gene, a big G and a little g. Say they got big G from mom and little g from dad. When they go on to produce their own egg cell here during the process of meiosis, then they'll pass on one or the other of those. So only one of those chromosomes makes it into the egg cell, and it's random, as we'll see. And in this diagram, I've shown you that the big G version makes it into that egg cell. So the chance of putting a big G into the egg cell is 1 out of 2 possible outcomes. So it could have been big G or little g, and the possibility of big G specifically is 1/2, 1 out of 2. Now, just to complete the life cycle, let's suppose that this egg cell that has a big G gets fertilized by a sperm cell that has a little g, and that would perpetuate this as a cycle. But of course, every generation doesn't have to have the same genetic makeup as its parents. So we're going to see some variation on this. Okay, so G, you look at another chromosome with another gene. This is one that's also got two versions, big D and little d. And let's say in this case that the little d ends up in the zygote there as a result of meiosis. I'm sorry, little d ends up in the egg cell as a result of meiosis. And that would be 1 out of 2 again. So 1 out of 2 possible outcomes, 1/2 chance. And let's say we were looking at G and D together, you could investigate the chance that this outcome happened, big G and little d together, 1/2 chance for big G, 1/2 chance for little d combined together. We would have to maybe look at this in a little more detail. So let's say we have a parent that's got two versions of G and two versions of D. And we want to know the possibility that a gamete has big G and little d together. That was the one, right? Big G and little d. So a few different ways to look at this. I'm just introducing you to the concepts right now. We'll get into a little more mathematical version of this as we go. One way to look at this is that we're combining two outcomes together. 1/2 chance of producing big G in the gamete, 1/2 chance of having little d in the gamete, and multiplying those together, you get 1/2, 1/2 equals 1/4. Another way to look at that is to consider all possible ways that these alleles could line up in the gamete, and shouldn't be too hard to understand that you could get 4 possible combinations in the gametes. And it turns out that those are all equally likely. So 4 possible outcomes, all equally likely. That gives you a 1/4 chance that each outcome would happen, right? So there are 4 outcomes, any one outcome, 1 out of 4. So this will be something we review a few different ways. You can extend it out into a larger number of genes. So let's say we've got this combination here, 2 kinds of B, 2 kinds of Q, and the same kind of N. So big B, little b, big Q, little q, big N, big N. So let me get some terminology out of the way, I guess. So when we talk about the versions, the alleles that are present in the individuals, that's called the genotype. And you could talk about the genotype of an individual who's diploid or the genotype of a gamete. And let's get back to that set of chromosomes, 2 B's, 2 Q's, one type of N. So if we wrote the genotype of that individual, 2 kinds of B's, 2 kinds of Q's, one type of N. And the convention is to write them with the capital letter first. And that allows us to line them up in a way that would be consistent regardless of which parent they get their B's and Q's from. We've also got, I guess, another terminology that I haven't introduced yet. I'm sure you've seen it before. A couple of these genes are what we call heterozygous, meaning that they have 2 different alleles. Heterozygous. So hetero for different. And the N gene in this case is what we call homozygous. It's got 2 of the same kind of alleles. So you can be homozygous dominant or homozygous recessive. This one has big letters, means they're dominant. We'll talk about that later. For now, we'll just talk about them as being homozygous. Okay, so how many possible combinations here? We've got B, big B or little b, Q, big Q or little q, and then with the N, there's only big N. So we can kind of take a different approach to the N genotype relative to the B and Q genotypes. So we want to ask the question of when this individual goes on to produce gametes, what are the gametes going to look like? So we could write those out. Big B combining, I'm going to do the first 2 letters first because that's going to be familiar with what we've done already. So big B can combine with big Q or little q. Little q can, or sorry, little b can combine with big Q or little q. So we still have these 4 possibilities. What's going on with N? Well, there's only one kind of N. So we don't have to complicate things anymore. We can just tack on a big N to all of these and find out that there actually are 4 possible combinations, right? And this is something that I really like to encourage in this class. You might have had a really like a version of doing this that you didn't think about too much. If you think about this, you save yourself about half the writing. So if you wrote too many big N's, more than you need to, you'd be writing double. We'll try to save some time on that in this class. So you could try to figure them out just by kind of eyeballing them that way. There are lots of ways to put these together. And this is one way to consider the ways to make gametes. So this is what's called a branch diagram. And it's similar to what I was doing, except it's got blocks as you go from left to right. We consider one new gene every time. And the blocks are indicating the chances of different alleles. There are 2 kinds of alleles for B, and there's a half chance of the gamete getting either one of those. So big B or little b. And then as a branch diagram, within the big B, independently, it can get big Q or little q at 50% chance. If a gamete gets the little b, it also could get the big Q or the little q at 50% chance. And that gives us now 4 blocks of breakdown, the way the B's and Q's can combine. And then this N block, just translate everybody straight across into 100% chance they get the big N allele. And that does just what I did on the other diagrams. So gametes, 4 possible gametes, big B with big Q, big B with little q, little b with big Q, little b with little q. Kind of glossed over what was shown on the previous slide. There's a way to write the genotypes that's a little more technical. We're going to use this in some contexts where we combine these with a slash between alleles of the same gene. and a semicolon separating one gene from the next gene. So this is the diploid genotype of the parent, big B slash little b semicolon, big Q slash little q, semicolon, big N slash little n. And that's diploid, 2 alleles for each gene. The gametes get 1 allele for each gene. That'd be big B, semicolon, big Q, semicolon, big N, and so on. So maybe start getting familiar with that notation. We'll use it sometimes, especially when we talk about the fruit fly genes in lab. Another way, so we did the branch diagram there. Another way to figure out your allele combinations is to do what I like to call a gamete square. So it's very similar to a Punnett square. Some people have learned this before in high school. I hesitate a little bit to show it to you because it looks a lot like a Punnett square. But in all these cases, as long as you know what you're doing and you keep track of that, you can use these if they're helpful to you and make sense. So what this does is it combines these independent chances of 50% big B or little b with 50% big Q or little q. And you just follow the B's down and the Q's across, and you get those 4 possible genotypes for B and Q. And combining those with N, we only have to write one kind of N because there's only the one kind of N allele, and that fills down big N for everybody. So another way to do that, whatever is easier for you. Some people are comfortable just thinking through how to make these gametes in their head. If you're not quite certain about what to do, I'd recommend using one of these methods, the gamete square or the branch diagram. All right, so this gets at a couple of Mendelian laws. We'll talk about these later, but this is the sort of principles of inheritance. And one of the neat things I like about genetics is that you can put this mathematical certainty onto some genetic features, such as the chance that big B or little b ends up in a gamete. So that 1/2 chance is a realistic expectation for how likely a gamete is to be made with one or the other of those alleles. So that 50-50 chance, that's what we call a genetic law, known as segregation of alleles, spelled out here. For any one gene, each of the 2 alleles has equal chance of ending up in a gamete. Another law here, known as independent assortment, says that the chance that a B allele ends up in the gamete is independent of the chance that a Q allele ends up in the gamete. So if we look back over here to what we did previously, we determined that there was a 50-50 chance of big B or little b, and out of 4, 2 are big B and 2 are little b, 4 possible gametes. We determined there was a 50-50 chance of Q, big Q or little q, and we see 2 big Qs and 2 little q's. So there's still equal chance that those are getting combined, and the chance that big Q ends up with big B is independent or equally likely that big Q goes with big B as big Q goes with little b. So that's the law. That's the law. Independent assortment. So a couple terms that are useful as you think about this, these inheritance patterns and predicting inheritance patterns in genetics. All right, so maybe one to think about. You can pause the video and try to work your way through this one. It's a set of 4 genes, 1 homozygous, 3 heterozygous, and you could solve this problem and try to figure out what the possible gamete genotypes would be and how likely each one would be to be made.